as you may know, circulating nucleic acids have been applied for a range of problems in cancer, including disease detection and monitoring, with a large focus on DNA mutations as informs, let's say, molecular residual disease, minimal residual disease. This abstract was a little bit different in that it focused on use of cell-free DNA, not for mutations, but for epigenetic changes that could be used to infer gene expression using a technique we described last year called EPIC-Seq, leveraging DNA fragmentomics at the start of transcription of genes of interest to infer their gene expression. So this study focused on design of a panel for doing this for lymphoma classification, focusing on 11 lymphoid malignancies, and the abstract presented how that design was performed and executed, and then separately an application of the EPIC-Seq targeted panel for this problem to two questions. The first was cancer classification for a range of lymphoma histologies: large cell, Hodgkin, follicular, mantle cell, CLL, transformed follicular, and myeloma. And the second question was around predicting a gene expression signature associated with double hit or cytogenetic aberrations of MYC and BCL2 as so-called the dark zone signature or the double hit signature. Both parts of the study were rather successful in the application of the method for classification. So in the method could distinguish patients with and without cancer, and looking at about 400 samples from patients with and without cancer, meaning lymphoma or non-lymphoma. And then more importantly, had a pretty high precision or positive predictive value for distinguishing lymphoma subtypes, including some challenging subtypes like transformed follicular lymphomas, where the positive predictive value was about 86%. And in some histologies above 95% positive predictive value. So that's a proof of concept of the study in showing that lymphomas can be classified non-invasively from the blood, but of course more work remains. The second part was focused on this dark zone signature. This is a subset of germinal center lymphomas that tend to behave badly, in large part because the foot is on the gas with MYC, and the brake is broken with BCL2 being rearranged. So this is a B cells out of control. And for us to measure them being out of control, we can, of course, rely on genetics, like the FISH tools that we have for finding these broken oncogenes. But of course, there are patients that don't have these oncogenes broken, but still have the same phenotype, a high gene expression signature. So we applied the same tool to resolve that in a group of patients, and we're surprised to see that this, what you might consider subtle distinction, was also discernible from plasma. You could distinguish patients with and without double hit by inferring the expression of the associated genes. So that was quite exciting to us.